Okay.
Perfect. So thank you very much for that. We continue with large-scale genomics. Andy Yates, refget overview. Andy Yates from EMBL-EBI. So, there we go. Oh, sorry.
Okay. I hope this is going to work. Fantastic. So, good morning, everyone. I am representing two work streams. I'm actually one of the co-leads of the Genomic Knowledge Standards work stream. I'll be presenting refget, which is, as has been previously described, an API which underpins the CRAM format. And very much as Mike was talking about before, about HSGet being one of these APIs that abstracts us away from traditional file systems, refget does the same thing for reference sequences. The first question might be, what is a reference sequence? I consider it to be a baseline of our knowledge. As the video this morning said, we have the Human Genome Project. It's a baseline of our interpretation, our knowledge. In order to make sense of other people's genomes, we have to have a reference point, somewhere that we can ground ourselves. And the reference genome and reference sequences can provide that grounding. Certain uses that we have for this is things such as aligning sequencing reads, calling variation, gene annotation, variation interpretation, and there are different types of reference sequences as well. Genomic, mRNA, cDNA, CDS, peptides, all of these are used commonly within bioinformatics analysis pipelines. Primarily, I'll be concentrating on genomic reference sequences during this talk, though. So how does reference sequence get used in a genomic analysis? A quick Google brought up a nice GA4GH germline caller, and I just highlighted the star here where it says map to reference. So that's the first place we're talking about. Actually, the very first step of doing any genomic analysis is mapping to a reference sequence. So we are really core. But actually, this completely underpins the entire pipeline as well. At the very end, you have a VCF that contains your variants. Variation is defined as a difference from the reference. So again, without a reference, you don't have variation. Now, another observation of doing sequencing and aligning that to the genome is that there's a lot of redundancy within the reads that align to the genome. This is an example of the Illumina BodyMap 2.0 RNA-seq experiment. It's a screenshot from the Ensemble browser, which I'm involved with. And I'm just going to highlight this region here where you can hopefully see a bunch of red little base pairs, and that's the only difference in this entire view. So there is a huge amount of redundancy in the reads. Now, observations, which basically led into the development of the CRAM format, says, well, if you can store those delta differences between the reads and the underlying reference sequence, then you can save a huge amount of space. So fantastic. What happens if you get the wrong reference, though? So here's a toy example. At the very top, we have a 5 base pair read. 3 of those positions are marked with an R. That means reference. The 4th position is marked with an A. So effectively, we said bases 1, 2, and 3 are referenced. 4 is a difference. Base 5 is reference again. The top genomic sequence we have here is GA4GH, which is the current reference human genome. The second sequence is GA4GH, which is the previous iteration. And the final one in here, which I chucked in, was the latest version of mouse. And if you go down, you can quite clearly see that if we look at the first read, actually that A is the same as the reference. So actually there's no difference. That cannot be the reference sequence. The second one, there is a difference. So we can assume that that was actually the genome in question that we aligned to. But also, if we look at the third sequence there, the read is completely different. So if we can't name chromosomes and we can't map back to chromosomes and we use this reference sequence, we use just the names. So this is all the same region. This is all chromosome 13. It's all the exact same region on that. We will reconstruct the wrong kinds of reads. So we really need to get this right in order to get the right kind of variation called. So how can we improve this? Well, you might go, well, the first thing is that we all agree on the same name. And we've got Keith here that says that scientists would rather share each other's underwear than use each other's nomenclature. Somewhat kind of true. So, okay, we do share the same names. We do talk about the same things in the same way, but we don't completely share that same nomenclature. So the second idea here is that we use a pre-existing universal naming system. INSDC already has this. But You might consider this as like one of those 416 scams. A genomic sequence reports to BCM000663.2. How do you know that? Oh, well, there's a header at the top that tells me that. No, really, how do you know that? How do you know that this really is a friend that's one of the military forces stranded in, that's currently stationed in Iraq? You don't know. Just because something says it is, doesn't mean it truly is. So actually, what we need is a way of actually verifying and identifying that that space actually exists. Now, of course, there's another problem here, which is naming as well. So there are a few problems in computer science. Originally, this was credited to Phil Carlton, who was a Netscape engineer, slightly modified for bioinformatics. 2 problems, off by 1 errors, cache invalidation, and naming things as well. So again, another reason why we should avoid using the names. So what can we use? Well, we can go back to computer science. We can say, okay, what do you have available for doing this? And there's something called checksums. And checksums are small algorithms which basically come out with a small identifier at the end that can be used to verify the data integrity, but you can't use it to verify data authenticity. I'm really concerned about integrity, so great, we can use that. And here's a little example of the idea that we apply to this. We have reference sequence, we normalize that sequence, we calculate a checksum, and out pops this string identifier, and that's a unique identifier for that given sequence. So refget is conceived as a method to retrieve sequence by a checksum. We also add in other sections in there, which involves being able to retrieve subsequences of that reference sequence and returning back key metadata such as known aliases. So again, is it called CHR1? Is it called 1? Is it called that CM number? Also the length of the chromosome as well. Now, the API is not limited to genomic sequence and chromosomes. Proteins and transcripts can easily be referenced in this, and there are also reference sequences, and the different refget deployments can choose what they want to provide. The idea is that the checksum that we use is sufficient to provide uniqueness across that entire set, and if anyone is thinking MD5 has collisions, don't worry, we'll come on to that in a second. This is also based on work from the CRAM project and the CRAM reference registry, as Thomas so elegantly put forward in the previous demo. So this is the implementation. We have a reference sequence. We normalize it by only allowing characters from A to Z. We uppercase that sequence and we allow no white space. From that, we can calculate 2 checksums. The first is based on MD5. The second one is called trunk 512, which is the SHA-512 algorithm that has been truncated to the first 24 bits of that digest, which is based on work conducted by Reese Hart as part of my work stream in the GKS work stream. Once you have these 2 identifiers, these go into a database, and then the API comes in with this URL sequence and the checksum identifier. It looks at what that sequence is in the background and returns back the sequence in question over a RESTful API. So an example, my examples are always done on small sequences like yeast, because yeast is small and it costs a lot less time to transfer an entire yeast chromosome over the wire than it does the whole human genome or anything like that. So we have this identifier here. I'm not going to say it, but it's the one starting 6681. That's chromosome 1 from Saccharomyces cerevisiae from the latest assembly. And there's these 2 URLs, which are the 2 live implementations of refget. The bit that's bolded is the identical URL. It's just the prefix that differs, and both return the exact same sequence. The same is true of the metadata endpoint. So again, we're querying the exact same sequence. The bits that are bolded in the response are the bits that are the same. So length and the MD5 is the same, but the 2 different services have decided to implement different parts of the metadata API. And this is completely allowed because we don't assume that every single API is going to be sufficiently complete in the exact same way. We understand that APIs will differ in their implementations. So in terms of the deployments and interoperability, the specification is available under the HCS specs repository. Deployments, we have a Perl reference implementation. The CRAM reference registry is the version that has all the data in there. And we're currently developing an AWS serverless model as well, which uses S3 Amazon buckets and also the Lambda function as well to provide the lookup functionality. In terms of interoperability, we took a complete queue from HCS get. We have our own specification for this, and we also have a compliance report, which looks a bit like this. If you're green, you're serene. If you're red, you're not. Actually, the endpoint that's failing there for the EBI endpoint is a tiny one. The service is absolutely fine. But if you were an implementer of this API, you can drill down into that information, find out more information about why the tests are actually failing as well. So the future directions for the API, there's two major directions as far as I'm concerned. The first is the observation that no sequence is an island. We do not perform genomic analysis on a single chromosome. We perform it on a huge range of these. So we need to somehow bundle genomes or transcriptomes or proteomes together, which basically means that we have to come up with a way of identifying collections of these reference sequences and be able to reference to that, and that gives redirections into these refget APIs for those sequences. The other question as well is we're looking towards the cloud. We want to distribute this across the cloud. It's really important, I see, for analysis being performed in the cloud that basically they don't come back to EBI for all the reference sequence. They go around to the local storage engine. The problem there is how do we federate across those instances as well? And this is another open question, both of which I think the discovery workstream have a part to play in that. So in summary, refget provides access to reference sequences, subsequences, and sequence metadata through a standard REST API. It's a foundational API of Global Alliance. It's one of the raw ingredients of genomic analysis as well. And our future work is going to provide robust mechanisms to define reference analysis datasets and networks to operate those APIs within. And with that, I just want to acknowledge everyone that's been working on the specification here. Many of them are not in the audience at all, but I do thank you. So thank you very much.